Good morning, students. First of all, I would like to brief up the syllabus of class 12 biotechnology. There are two units, and in each unit, there are three chapters. We will start with Unit 6, Chapter 3, Animal Cell Culture and its Applications. These are the subtopics of the chapter that will be discussed. Advantages and disadvantages of animal cell culture, features of animal cell in culture, types of cell culture, types of cell lines, physical environment for culturing animal cells, medium used in animal cell culture, equipments required and the growth curve of cell lines, methods of gene delivery, storage and revival of cells, applications of animal cell culture and hybridoma technology, stem cell culture, and tissue engineering. What is the need of cell culture? It is generally used for studying basic cell biology, for the interactions between disease-causing agents and cells. It is used for studying the effects of drugs on cells, the process and triggering of aging and nutritional studies. Is generally used for stem cell culture and tissue engineering. What is the need of cell culture? The cell is generally used for studying basic cell biology, interactions between disease-causing agents and cells. It is used for the effects of drugs on cells. It is used for the process and triggering of agent and nutritional studies, stem cell culture, and tissue engineering. The history of animal cell culture dates back to 1856 when Ludwig maintained animal organs alive outside the body by pumping blood through them. This is considered as the start of animal tissue culture. The cell culture was first successfully undertaken by Rose Harrison in 1885. For the first time, embryonic chick cells were maintained in a cell culture. In 1950, George Gay established the first human line in culture. In 1980, CHO cell lines, CHO means Chinese hamster ovary cell lines, were used to produce erythropoietin, an important product introduced to pharmaceutical market. Cell culture is defined as the growth of cells dissociated from the parent tissues by spontaneous migration or mechanical or by enzymatic dispersal under in vitro conditions. Now, what is this spontaneous migration? Spontaneous migration means during morphogenesis, the cell can separate and also during wound healing. And in certain cases, you will find the invasion of cancer cells. The second is the mechanical method of separating cells. For isolating cells from the intact tissues, first the extracellular matrix holding the cell together has to be disrupted. These are done by various forces. The first type is the collision forces. That means by subjecting the cells to liquid flow or exploding the cells by using pressure differences. The mechanical force is followed by proteolytic enzymes. The main enzymes that are used for separating cells are trypsin and collagenase. There are two types of cells as you know, that is eukaryotic cell and prokaryotic cells. The eukaryotic cells are more difficult to culture than prokaryotic cells. What is the reason behind it? It is the need of a very complex media, and it is very susceptible to contamination, that is due to the overgrowth of microbes such as bacteria, yeast, or fungi. Animal cells will continue to grow outside the living organism if supplied with appropriate nutrients and growth conditions. The process of growing of cells under laboratory conditions is called cell culture. The animal cell culture can be classified into three types of culture. The first is the organ culture, tissue culture, and cell culture. We will come to the organ culture. In organ culture, the entire embryos, organ, or tissues are excised from the body either by vivisection or shortly after brain death. What is vivisection? Vivisection means the practice of performing operations on live animals for experimental purposes. In this culture, the physiological functions of the organs are maintained, but the scale up is not recommended. The scale up means the development of culture system from small scale to large scale industries. Coming to the next one, that is the tissue culture. When fragments of excised tissues are grown in a defined media, it is called tissue culture. In this, the original organization of the cell is lost, but the as compared to organ culture, it is better for scale up, but not ideal. Then coming to the third type of culture, that is the cell culture. When the excised tissues are dispersed into a cell suspension and then cultured as monolayer, is termed as animal cell culture. This can be maintained for a very long time and it can be scaled up. The common theme to all cell culture. The cell density must be controlled. The cell density needs to be high enough to maximize the production of protein products. But if the cell density gets too high, the nutrients get depleted and the waste product starts to build up in the media. In the case of some cells, the cell-to-cell contact can cause cells to stop dividing, which is called as contact inhibition. What are the features of animal cell in culture? It needs to be replenished periodically. There is limited growth, even if the best nutritive and physical conditions are maintained. There are limited number of generations. Once the cell spreads to the entire surface and comes in contact with the walls of container, the cell stops growth and this phenomenon is called contact inhibition. The cell-to-cell interaction and the cell matrix interaction are reduced. The cell proliferation mechanism also changes. What is cell proliferation? It means to grow or multiply by rapidly producing tissues. Now there are certain advantages and disadvantages of animal cell in culture. So what are these advantages? It is easy to add genes. There are homogeneous genetic population, controlled physico-chemical environment, easy production of biopharmaceuticals, no ethical clearance required, cost-effective screening assays. But there are certain disadvantages. Since the animal cell is very small in size, high sensitive techniques need to detect the changes. The scale-up is very challenging and it may not represent in vivo phenotype or genotype. Let's now learn about various types of animal cell culture and the technology associated with it. There are two types of animal cell culture: primary cell culture and secondary cell culture. Coming to primary cell culture, the cells that are explanted directly from a tissue or organ of any donors and culturing it in nutritive media is called as primary cell culture. It is of two types: anchorage dependent cell and anchorage independent cells. So what are the features of anchorage dependent cell? These cells adhere to the bottom of a flask and form a monolayer. Eventually, it will cover the entire surface of the substratum and the growth stops. It needs to be subcultured and then again the proliferation can begin. Coming to the second type, that is anchorage independent cells. These cells are associated with the body fluid, usually the blood cells, and it is grown in suspension. It will eventually need subculturing. The drawbacks of primary cell culture are that they are time consuming and require the use of live animals or fresh tissues. There can be considerable variation from one preparation to another if prepared by different people. These difficulties can be overcome by the use of secondary cell culture or cell lines. So, what are secondary cell cultures? The primary cultured cells, when excised from the specific organs or animals, when subcultured in a nutritive media, is called secondary cell culture. The subculturing involves the removal of the cells from the primary media, washing the plates, and dissociating the adhered cells with enzyme trypsin and diluting it in fresh media. Sometimes certain cells from the secondary cells get altered and give rise to continuous cell lines. There are two types of cell lines: the finite cell lines and infinite cell lines, or the continuous cell lines. What are the features of finite cell lines? The finite cell lines grow through a limited number of cell generations and have limited life. It grows slowly and forms monolayers. The doubling time ranges from 24 to 96 hours. It is usually anchorage dependent. Contact inhibition and density limitation is usually seen. The cell lines, transformed under in vitro culture conditions, give rise to continuous cell lines. The various properties associated with continuous cell lines are: there is no contact inhibition, it is not anchorage dependent, the mode of growth is either monolayer or suspension form, the growth rate is rapid, the doubling time is between 12 to 24 hours, and the density limitation is reduced or lost. The culture should be examined daily for their morphology, color of the media, and the density of the cells. The cells are usually grown and maintained in petri dishes, culture flasks, or multi-well plates of various shapes and sizes. The culture conditions can vary for each cell type, and variation of the condition can result in different cells being expressed. A cell culture laboratory should have equipments like tissue culture hood, CO2 incubator, inverted microscope, centrifuge for doing animal cell culture. The basic equipment is laminar airflow. All tissue or the cell culture must be performed without any bacterial or fungal contamination. Otherwise, the animal cell culture can easily get contaminated with bacteria or fungi which will outgrow animal cells. The laminar airflow allows the work area to be free of such contamination. It essentially provides two functions. It protects the tissue culture from the operator and also protects the operator from the tissue culture. Depending on the nature of the cells or the tissues being handled, the biological safety cabinets are designated as Class 1, Class 2, and Class 3 laminar airflow. In Class 1 laminar airflow, maximum protection is seen and the inward flow of air can contribute to contamination of samples. In Class 2 laminar airflow, it provides both kinds of protection of the samples and that of the environment, since the makeup air is also HEPA filtered. What is HEPA? HEPA means highly efficient particulate air. In Class 3 laminar airflow, the cabinet is installed in laboratories where maximum protection is required. It is specifically designed for work with pathogenic agents and it provides maximum protection. Inverted microscope. In animal cell culture, the cells are present at the bottom with the culture media above. The inverted microscope allows the cell at the bottom to be visualized because the optical system is at the bottom with the light source on the top. The microscope should be kept covered to protect from dust and the light should be turned down when not in use. Various types of sterilizer equipments are used. Of that, autoclave is a must in all laboratories. Then coming to the low speed centrifuge. A low speed refrigerated centrifuge is generally used for the isolation of the cells from suspension cultures. What is the reason for using low speed centrifuge? The spinning of the cells at very high RPM can result in the breakage of cells. In ordinary centrifuge, the temperature can increase and at high temperature the cells get damaged and further metabolic activities will not take place. Then coming to the most important type of equipment used in animal cell culture is the CO2 incubator. The CO2 incubator is designed to reproduce as closely as possible the environmental conditions of the animal cells. The essential functions of the incubator are to maintain the sterility of the chamber, a constant temperature, an atmosphere with fixed level of CO2, and high relative humidity. The animal cells are grown in an atmosphere of 5 to 10% carbon dioxide because the media used is buffered with sodium bicarbonate or carbonic acid, and the pH can be strictly maintained, and it also maintains the correct osmolarity. The culturing of animal cells under in vitro conditions involves the creation of appropriate physical, nutritional, and hormonal environment in which the cells can grow. The physical environment includes the controlling of the temperature, pH, osmolality, and gaseous environment by providing a supporting surface and protecting the cells from chemical, physical, and mechanical stresses. The most important physical environment that has to be maintained is temperature. The mammalian cells are grown in incubators maintained at 37 degrees centigrade. This temperature is usually maintained because the body temperature of the Homo sapiens is 37 degrees centigrade. Further, it has been observed that most cells derived from the warm-blooded animals also grow at this temperature. The next physical environment that has to be maintained is the pH. The regulation of extracellular and intracellular pH is essential for the survival of animal cells. The correct pH for maintaining the cell culture is 7 to 7.4. The regulation of the pH is done by using a variety of buffering systems. Most media use the bicarbonate CO2 buffering system. Why pH is important? It usually maintains the appropriate ionic balance but also maintains the optimal function of cellular enzymes and also for optimal binding of hormones and growth factors to the cell surface receptors. Coming to the next physical environment is the osmolality. The osmolality of the culture media has a significant bearing on cell growth and function. It is generally used to preserve the membrane integrity of the cell. If the outside osmotic pressure becomes higher, then the cells will shrink or swell accordingly. The osmolality of the media is determined by the media formulation. The salt and glucose are the major contributors to the osmolality. It is generally checked directly by using an osmometer. The rest part of the chapter will be continued in the next video. Thank you.